I'm going to be recording. We're welcoming, this is a special ALK talk where we have Dr. Ben Solomon, who lives across the world in Australia. So right now it's Monday, I believe Monday morning for him. And he's taken out an hour of his clinic to talk to us, which is amazing. And how fortunate that we're able to speak to a world-class doctor that specializes in ALK-positive. And I want to thank Dr. Solomon for coming. Next Sunday, we're going to be kicking off ALKCAM, Lung Cancer Awareness Month, for the month of November. And we're going to have Chris Traff and others at a regular 7 o'clock Eastern time. And I will turn it over to Jeff Strum. Jeff Strum is a member of our medical committee, and he will be monitoring the talk with Dr. Solomon. Thanks. Jeff, it's all yours.
Greetings, everyone, and welcome to ALK Talk. Today's discussion is about the investigational drug TPX-0131, developed by Turning Point Therapeutics, Inc., and about the FORGE-1 phase 1/2 clinical trial, which is ongoing both in the US and Australia. Joining us today is Dr. Ben Solomon, principal investigator of the trial at Peter Mccallum Cancer Center in Melbourne, Australia, where it is 8 a.m. Monday morning, and Dr. Solomon is getting ready for a busy day with patients. I am Jeff Strum, a 7-year ALK+ survivor, currently on lorlatinib, and a member of your ALK-positive clinical trials and research acceleration committees. TPX-0131 is the first 4th-generation tyrosine kinase inhibitor for ALK-positive non-small cell lung cancer to enter the clinic, and represents a great source of hope for ALK patients, particularly for those of us on lorlatinib that may eventually regress with certain single and double mutations. There have been initial reports about a rough start for the trial, which is a first-in-human dose-finding and initial efficacy study. While the company and collaborating investigators are prohibited from talking about specifics of the trial in the US by the FDA, HIPAA, and SEC and other regulatory bodies, Dr. Solomon has agreed to talk with us in general terms that will broaden our perspective and knowledge of how clinical trials are designed and structured. Dr. Eric Park of Turning Point could not attend due to regulatory constraints and concerns of his compliance officer. Rather than proceed with our usual open forum for questions today, your clinical trials and ALK Talk committees decided to gather our own questions internally so as not to push the boundaries of privacy and disclosure regulations. With that, I'd like to introduce you all to Dr. Ben Solomon of the Peter Mccallum Cancer Center in Melbourne, Australia. Good morning, Dr. Solomon, and welcome.
Good morning, Jeff. Good morning, everyone. Thanks very much for the invitation to chat to you all.
Before we proceed with your presentation, can you tell us a little bit about your background and particularly how you came around to doing research into ALK-positive non-small cell lung cancer? You were involved in the trials of other ALK TKIs, correct?
Yeah, sure. So so just by way of background, so I'm a thoracic medical oncologist. I did my training in Melbourne, but then I did a 2-year postdoc in Colorado. And then I came back to Melbourne in 2006. And just about then, at the end of 2006, the very first phase 1 trial that I did was a trial with a drug called PF02341066. Now, you may not have heard that number, but that drug was a drug which we all know now as crizotinib. Crizotinib, of course, was the first ALK inhibitor, and it was an amazing trial. We learned a lot about targeting ALK, ROS1, and even c-MET. So since then, I've been involved in a number of different trials with targeted therapies, and in the context of ALK, we've been involved with phase 1s with crizotinib, ceritinib, lorlatinib, and we'll be talking a bit about a new phase 1 trial which we've just opened up. So Jeff, were you happy for me to sort of start my presentation?
Yes, please do. Thanks.
Sure. So I'll just try to share my screen. Bear with me. All right. Okay. So just by, this is my disclosure slide. I note that I have not received compensation from Turning Point Therapeutics for this presentation or really any other purposes, and that some of the products, in particular TPX 131, are investigational and not approved for use by any health authority in any indication. So I mentioned this drug crizotinib, which was the first-in-class ALK inhibitor, and way back in 2011, we showed that crizotinib was active in tumors with ALK rearrangements. And then we went on to do a phase 3 trial that showed that anyone who was had a newly diagnosed ALK-positive lung cancer should be treated with an ALK inhibitor rather than chemotherapy. But it's been an amazing field because it's moved on with the development of new and actually better drugs, first generation, second generation, and third generation ALK inhibitors. And what all these drugs share in common is that they're more potent against ALK. They have better coverage of mutations that confer resistance to crizotinib and earlier generation compounds, and they have better CNS penetration. So the concept of resistance is an important one. These drugs work very well, but tumors can develop resistance to ALK inhibitors. And these are the sort of major mechanisms by which tumors become resistant. Sometimes it occurs because tumors in the brain progress, and this can be related to lower concentrations of drug in the brain than outside the brain. Or it could be that the tumor cells become rewired to make the actual tumor cells stop being inhibited by the ALK inhibitor. And probably the best characterized mechanism responsible for this, a development of mutations in the rearranged ALK gene. So we know that the type of mutation that you get after different ALK inhibitors is different. So with crizotinib, you get a particular spectrum of mutations. With second generation inhibitors, you get a different spectrum of mutations. These mutations tend to be single mutations, and in the context of the second generation inhibitors, the G1202R mutation is probably the most common. I'll briefly talk a little bit about lorlatinib because I think it provides a bit of context to this, to the discussion that'll follow. So lorlatinib is a bit different to the second generation inhibitors. It's got a more compact structure, which is called a macrocyclic structure. It's the macrocyclic refers to, I think you can see a circle within the structure. And that makes it a more compound molecule, which is able to cross the blood-brain barrier more effectively. And it's also active against mutations, including the G1202R mutation, which results in resistance to some of the other inhibitors. So the lorlatinib study, phase 1/2 study, had two parts. It had a phase 1 part. And in the phase 1 part, the key part of that was to determine the safety of the drug, but also to work out what dose was required to move into the phase 2 and ultimately the phase 3 part of the study. A dose of 100 milligrams was chosen, and we learned a lot about the safety and side effects of the drug, and and learned how to manage these side effects. Once we did that, we were able to open the part 2 of the study, which had these different cohorts looking for activity in different groups. As a result of several of these cohorts, lorlatinib was approved in patients whose tumors had progressed after one or more ALK-tyrosine kinase inhibitor. And the subsequent phase 3 study, the CROWN study, has led to approval in the first line as well. And these are just some of the data from the phase 2 study with activity in patients in tumors both outside and outside the brain and also in the brain. And lorlatinib, importantly, was able to overcome resistance due to this G1202R mutation. But we know that tumors can become resistant to lorlatinib. And this is work from Alice Shaw's group from the Mass General Hospital. And the patterns of mutation responsible for resistance are a little bit different. I talked about those single mutations, but what we see emerging with lorlatinib is these, what we call compound mutations. And so a tumor develops resistance to a second generation inhibitor with a single mutation. Then we start treatment with lorlatinib, and then a second mutation occurs together with the original mutation, and that causes resistance to the lorlatinib. So these compound mutations, we know from the lab, are very hard to inhibit with the existing drugs that are out there, including lorlatinib. This has been seen by other groups, and the emergence of these mutations can be tracked in plasma. And similarly, this group also showed in the lab that the compound mutations were resistant to lorlatinib and the existing ALK inhibitors. And this is work from Ibiai, Dagogo, Jack, looking at both plasma and tumor biopsies post-lorlatinib, showing that you frequently detect more than one mutation in the context of progression. So we do need drugs that are able to deal with these mutations. So that sort of, that brings us on to the discussion about this new molecule, TPX-0131, and the phase 1/2 trial, which is called FORGE-1, which is designed to test the safety and activity of this drug. So this is a little bit like the picture I showed you about lorlatinib. So TPX-131 is a newer generation, highly potent ALK inhibitor. Again, it's got this compact macrocyclic structure, and it's actually a bit more compact than lorlatinib. And it's able to get into the brain very effectively in preclinical models. And it's active against a broad array of mutations, including the solvent front mutation, the gatekeeper mutation, but importantly also compound mutations. So this cartoon speaks to how inhibitors work and how mutations can cause resistance. So tyrosine kinase inhibitors work by stopping ATP entering what we call the ATP binding pocket of a kinase. They sort of lock into this pocket and prevent ATP from getting in. But if you get mutations, and these can be either at the solvent front or in a different position, the so-called gatekeeper position, that can prevent these molecules from getting in. And if you get more than one mutation, it can be particularly tricky for an inhibitor to enter that pocket. And so this molecule, because of its compact small structure, is able to get into places that the second generation inhibitors or lorlatinib can't get into, and therefore is, at least in the lab, able to have activity against single and compound mutations. So there are lots of numbers here on this table, but I think there are a few important points to make out. So this is a table which shows different ALK inhibitors and how effective they are at inhibiting ALK, wild-type ALK, or an ALK with different mutations. And the key point is the lower the concentration that's able to inhibit the mutations, the more effective or more potent the drug is. And TPX131 is shown in the top row, in the row that's highlighted in yellow. So if we look first at wild-type ALK, so this is ALK without mutations. TPX has the lowest what's called IC50, so it's the most potent drug at inhibiting regular wild-type ALK. If we look at single mutations, it does seem to be, again, the most potent, but I do note there are particular mutations where some of the second generation inhibitors actually might be more potent. But I think this is probably where it really stands out in terms of the compound mutations. It really is by a long shot the most potent inhibitor of these mutations. It's important to know that these are studies done in the laboratory, actually in cells, and these findings really do need to be moved from the lab to the clinic, and that's the whole purpose of clinical trials, and that's what we'll be talking a little bit about. The people at Turning Point have shown that this work, which I showed the table, which is based on cell lines, does seem to hold out in mice. So if we look, these are curves of growth of tumors in mice, and the purple or the fuchsia curve at the bottom is TPX. And it's able to inhibit growth of not just tumors with single mutations on the left, but also tumors with compound mutations shown on the right. So that gets us to the design of, so I guess I've spoken to about what TPX131 does in the lab. But it's important to see how this translates to tumors in actual people, and hence well, that's the rationale for the trial. And we'll talk a little bit about the design of the trial. So the trial is designed as a phase 1 followed by a phase 2 clinical trial. So we call these phase 1-2 clinical trials, but it's important to note that the first part of the trial is a phase 1, and then from the phase 1, a dose is determined, the safety profile is established, and then the trial moves on to its phase 2 component. So this is a first time in human trial. The drug's been evaluated in the lab. There's been preclinical toxicology, which has been done in 2 different models. This is typical for most, pretty much every drug. Typically, the preclinical work's done in rats and non-human primates. And then the aim of this study is to evaluate the safety, the tolerability, the PK. PK stands for pharmacokinetics. That refers to the concentrations of the drug in the blood, and also the efficacy of TPX131 in patients with ALK-positive non-small cell lung cancer. And I talked about the 2 parts of the study, the phase 1 and the phase 2 components. So this is the design of the study. So there's an initial phase 1 component. And how phase 1 studies work is, although we learn a lot about the drug preclinically, we do need to see that the drug is safe in people, and work out what the right dose to use is. So a dose is chosen on the basis of what's known from the preclinical studies, and it's a dose which is expected to, which is predicted to be a safe dose, but as close to an effective dose as possible. But safety, I think, is the key component here. And the trial, and this is typical of most phase 1 trials, involves entering a small group of people at each dose level. Typically, this is a 3+3 design, which means that typically 3 people are entered on at one dose level. They're followed for a certain period of time. Then their toxicities are reviewed. And if it's determined that the drug is tolerated okay, and there's also other factors, drug levels are looked at, then the trial moves up to the next dose level. Now, it's also possible that that a drug may move down a dose level or enter a dose level that is in between predicted dose levels based on the protocol. So there's a lot that can sort of change in a phase 1, depending on what's observed and what's learned about the study. Then once a dose level is, once a dose is established, then the trial moves on to the expansion level. And just to sort of give an illustration, I talked about the lorlatinib phase 1. With the lorlatinib phase 1, I think the starting dose level was 5 milligrams. You'll remember that the ultimate dose was 100 milligrams, but we went right up to 200 milligrams and had to come back down to 100 milligrams. So there is a lot of learning that happens during a phase 1 study, and that's important for people considering participating in a trial, in a phase 1 trial, to be aware. So the objectives are listed there: safety, tolerability, and to work out the maximal tolerated dose. More importantly, actually, the recommended phase 2 dose to move in. It's also important to understand how the drug behaves in people. So in a phase 1 study, there are lots of additional tests in terms of things like ECG, we call them ECGs, I think you guys call them EKGs, but also lots of blood tests looking at drug levels to establish to work out what the pharmacokinetics is. Because we don't really know from the preclinical models how exactly the drug will behave in people. And in different phase 1s that I've been involved in, we sometimes find that even at the initial dose level, the drug can result in higher concentrations than we predicted, or alternatively with lower concentrations. And this is the only way we can learn this is through, really the very generous participation of patients on a trial. So these are some of the inclusion criteria for the study, and I think if people are considering participating in a trial, it really is important to talk to their docs about whether the trial is suitable for them. But there are certain inclusion criteria and exclusion criteria that are built into every trial. And part of the purpose of the inclusion criteria of the trial is not just to make sure that a particular patient is suitable for the trial, but also the reverse, to make sure that the trial is suitable for that patient as well. So it works two ways. Inclusion criteria and exclusion criteria protect patients as well as protect, as well as ensuring the integrity of the trial. So these are some of the inclusion criteria for this particular trial, which are pretty typical in terms of age, confirmation of ALK-positive non-small cell lung cancer. There is a requirement for measurable disease and good performance status, adequate organ function. The trial, and I think this is a good thing, does allow people with asymptomatic CNS metastases, including leptomeningeal disease. The trial criteria was amended with respect to number of lines of ALK tyrosine kinase inhibitor therapy. Initially, it was very restrictive, but this has been broadened to allow up to 3 prior lines of ALK-TKI treatment into the dose escalation. These are the exclusion criteria, which I don't think there are any sort of standouts there. It's important that people are well and of good performance status going into a trial. These are so in the very beginning of a phase 1 study, it's a bit different. In a phase 3 study, we have a good feel for the likely side effects of the trial, and in the information and consent form, you can have a very detailed list of what to expect. With a phase 1 trial, we really learning through the trial. There's very little information at the beginning of the trial. We learn as we go on. These are some of the potential risks based on the preclinical toxicology that have been seen. But I think this is something that we'll learn more as the trial goes on. The trial has started recruiting at several centers in the US. I think in addition to these, I think the Mass General is just about to open. And the trial is open at some centers in Australia as well, including our center at Peter Mac. And Jeff, I think I'll finish there if that's okay with you, and happy to chat further.
Terrific, Doctor. Thank you very much. Clarified quite a few things and answered a few of my questions. What would be the typical timeline for the completion of a TKI trial?
Gosh, so I talked about probably my favorite trial, which was the crizotinib trial. We had that open at our center for 10 years, Jeff. But that's a little bit of a flippant answer. The aim of a phase 1 trial is to learn as much as we can about the safety and work out the dose as quickly and as efficiently as possible, so we can move on into the sort of phase 2 and hopefully the phase 3 part. So typically that will take a year or 2 to work through that. And the reason for that is because there is a lot of, the absolute priority of the trial is patient safety. So I talked about having a group of patients that go on initially, they get followed, and then a decision is made about whether it's safe to go up a dose. And then the next group gets on, they get followed, and then that takes time. So it is hard to say, but the general principle is to get through it as quickly and safely as possible.
Is there any perspective on why there are such differences in the way people react to these drugs? I mean, some people have one reaction, some another, some are fine, some get very sick. Why is that?
Yeah, so that's another great question. I think in part that can be because different people have different, you can get different levels of drug in different people. So that's one possible explanation, that in a particular individual, for whatever reason, the concentration of drug in their system might be higher than in another patient.
Is that because their absorption is different? They absorb it differently?
Absolutely, it could be related to absorption. There are potentially ethnic differences as well. So a lot of you will be aware that the dose of alectinib in the Japanese population is actually half the dose in the Western population, but the drug levels are pretty much the same. But a lot of it is related to sort of just individual tolerance for reasons we don't fully understand. And we know that even with the drugs that all of you are familiar with, drugs like crizotinib, alectinib, lorlatinib, even in the phase 3 trials, about 10% of patients have to discontinue drug because they don't tolerate it.
So when starting a first-in-human trial, how is the dosage of the medication chosen? I mean, do you scale up proportionally from the weight of the mice, or how do you figure that out?
Yeah, that's a great question. So there is an element of scale up, but I guess the principle is to try to use, there's a tension between getting to an effective dose and the safety, and there's a balance there. So there are predictions that are made about what an efficacious dose is, but there's also close sort of monitoring to see at what dose levels toxicity was observed in the different models that we use, whether that's in rats or in non-human primates. And the starting dose is typically at a dose where very minimal or no toxicity was seen in the preclinical models, and then and the doses are stepped up from there. And that's something that the sponsors will do and will discuss with the FDA about how to do it and what dose to use, and where to start and how quickly to go up on the doses.
During preclinical studies, how are the side effects assessed in animals on the medications? I mean, you can't really tell if a mouse has a headache, I suppose.
Yeah, that's very true. So in the preclinical models, they actually do observe the animals, and I don't know whether they can sort of infer things about like headaches, but they can look for signs of distress in the animals. But they look at levels in the blood, and they actually do postmortems as well and examine tissues such as nerves, the heart, actually the brain as well. So learn as much as they can. But these, and it's a good point you raise, that there are lots of good reasons why these models aren't predictable, and we know that rats are not people, so they really just give us a place to start rather than tell us something too definitive.
Can the therapeutic dose be different from those originally outlined in the protocol? I mean, can you miss completely and have to cut it by orders of magnitude, or increase it by orders of magnitude?
Yeah, so a lot of effort goes to try to get it as close to to try to sort of minimize that. But yes, I think you learn as you go from the study. You learn from the drug levels, from all those any of the people on this this Zoom that have participated on clinical trials, particularly phase 1 trials, will know that they get a whole heap of blood tests for the PKs, and we learn from that about how the drug behaves in terms of drug levels. We try to understand. We learn what the peak concentration is, what the trough concentration is. And sometimes on that basis, we do need to change track, and that might mean instead of going up to the next planned dose level, we might go up somewhere in between, or we might learn that we can move quicker than we thought.
And what is the process of changing the study design? If the phase 1 portion shows that a revision would be beneficial. I mean, do all of the PIs get together and talk and discuss it with the company? And describe that again, if you will.
Sure. So I talked about the sort of different dose levels in the phase 1, and at the end of what we call so patients get enrolled on a dose level and get followed for a period of time, which is typically a minimum of 1 cycle. And there's certain toxicities that we look out for. These are called dose-limiting toxicities. At the end of that period, there's typically a meeting that's held between the investigators and the sponsor of the trial, where the experience of the patients, their toxicities, the PK, the plasma, the blood levels of the drug are reviewed. And then a decision is made at that point, and a recommendation will be made to go up to the next dose level, or to consider an intermediate dose level. Now, the wording of most phase 1 protocols is sufficient to allow that to be done without an amendment made to the protocol. But sometimes if the change is a very big change, then an amendment may be required, which will sort of add time to the process.
Yeah, that was my next question. When the need for dosage changes arises in a clinical trial, does that extend the expected completion date of a particular phase or phases?
Yeah, so it may, only because that might mean that there's sort of added uncertainty about the dosing. There've been trials that have been involved in where the trials moved from once, it was planned that it'd be a once daily dosing, but then it emerged that some of the toxicities might be better managed if it was twice daily dosing. And then we've had to explore twice daily dosing levels, and that has sort of increased the amount of time taken to get to to identify the recommended dose. But I think that is important. This is really the only opportunity to, well, it's not the only opportunity, but it's the most important opportunity to get the dose right. You don't want to start a phase 2 or a phase 3 and have to go back and think, have we actually got the dose right? Do we need to change?
How often, in your experience, have you seen the dosage drop significantly? They call them dose escalation. I, in my prologue, called it a dose finding study. Does it often go down?
Yeah, I do like that you call it a dose finding, because sometimes you go, you go up, and you might go up further than you thought. I sort of mentioned in the lorlatinib study, we went up to 200 milligrams, and I'm sure a bunch of you are on lorlatinib, and some of you have even had to go down from 100 milligrams to 75 milligrams. But the only way we learned that was through going up and then realizing we had to go down. And a lot of protocols have built in what we talk about a dose level 1, dose level 2, but there's often a dose level -1 that's built in to allow for that to happen. So it's possible.
We usually hear about maximum tolerated dose in clinical trials. In the clinical trial criteria, but is it at all common to be testing for a range of effective doses rather than simply finding the maximum tolerated dose?
Yeah, I sort of prefer to think about the aim of a phase 1 trial to find the recommended dose, or what's now called the recommended phase 2 dose. A dream scenario is that you get a drug where you can keep going up and you don't get dose limiting toxicities, and you're clearly in the range where the drug is effective and is able to do in people what it does preclinically. So you can have some trials where without a maximally tolerated dose, you identify a recommended dose. That sort of paradigm of maximal tolerated dose was a paradigm that was sort of developed in the era of chemotherapy trials, where you'd sort of push as hard as you could until you got toxicity, then you'd back off one level, and that would be your chosen dose. With targeted therapies and with our understanding of how they work, what concentrations are needed to block the target in tumors, we don't have to be as blunt as that, and we can use other information to work out what the recommended dose is. But to me, seeing tumors shrink is actually the best indication that you're probably at the right dose level.
When I was on, I'm going to go off the reservoir here for the reservation for a minute here. When I was on alectinib, I couldn't tolerate it. And I kept dropping the dose. I kept dropping the dose. And I called up Genentech and I asked them if they had an assay, if they could tell how much was in my blood. And of course they had one, but they wouldn't give it to me, of course. When you think about it in retrospect, I wound up going all the way down to 350 milligrams a day when the original was 1,200 milligrams a day, and I still could hardly tolerate it. And I was on that for an additional 3 years without any regression, 5.5 years. So something in me was a really highly absorptive of alectinib. And it seems to me that there should be a way to assess the level of these drugs in people's bloodstreams, and yet there isn't.
Yeah, that's interesting. Interesting you say that, and it is a little bit curious that we dose chemotherapy based on people's height and weight. Targeted therapies, we sort of say that there's one dose that'll suit everyone, and that's clearly a simplification. It is to do in part with the fact that we've been sort of lucky that there has been what we call a therapeutic index, so that for most people, even if you're a little bit higher than you need to be, it'll be tolerated well, and a dose is chosen that'll be effective in most people. But there's no reason to think that the drug levels will be the same in everyone, and I think your experience with alectinib speaks to that.
Yeah, one of the rhetorical questions that came my way from our group. The hope is that patients will be on these TKIs for a very long time. So how is the balance between efficacy and side effects to be maintained?
Yeah, that's absolutely the hope, that people will be on for a very long time. I think, I guess the first thing is that the best way of maintaining quality of life is to suppress the tumor and hopefully get rid of the tumor. But in addition to that, you want to have a toxicity profile that means people can be on it and in other ways maintain their quality of life. So that's a learning thing. I've talked about lorlatinib a few times. Lorlatinib is a drug which is very effective, but it does have side effects, and we've learned as we've gone about how to manage those. The cholesterol, for example, we know which drugs to use to lower the cholesterol and triglycerides. The cognitive effects, which can be problematic to a bunch of people. We know that dose reduction is very effective, and we talk to people and their family, to patients and their families about being pretty proactive about reducing dose. So part of that, so it's a learning thing, and not all the learnings will happen at the end of the phase one. I think you hope to get as many as you can, but we keep learning as we go. And it's very interesting that you spoke about alectinib. My pharmacist just emailed me to ask whether they'd be interested in setting up an assay for alectinib levels to finish dosing. So maybe we'll be doing that in the future with TKIs. But I guess the short answer to your story is we're not going to have all our answers at the end of the phase one. We keep learning.
We have 2 more topics, 12 more minutes, exclusions and washouts. So why are exclusions like the one in the Forge 1 entry is currently limited for patients that have experienced up to 3 prior TKIs and 1 line of chemotherapy? Now, the ALK-positive, as you may know, collaborated with Turning Point to get it raised from 2 prior TKIs to 3 prior TKIs. But we have PIs here in the United States that are concerned that that particular exclusion knocks out some of their patients that have 3 prior TKIs and 2 chemotherapies, for example. And there's a significant population of those that are getting to the end of the road that really need this particular drug. Why are exclusions like this included in the trials? And are relaxing those kind of exclusions down the road a possibility?
Yeah, no, so that's a great question, and I have to say on that specific example, I completely agree with the views of this group, and I welcome the sort of the input of this group in their discussions with Turning Point, but I have to sort of also let you know that a lot of the investigators provided that feedback to the company, and I think the company were influenced by our as in investigator feedback, and also the feedback of your group, and made that modification. So I guess it is a dynamic process, and yes, as as the company learns, and hopefully we'll see activity in patients with with 3 prior TKIs, and I think that'll make them feel more comfortable about opening up. But to your question, why do they even have these restrictions in the first place? I think, again, that's sort of probably more a question to address to them. But if I had to think about why they would, a lot of it comes down to they need to get the trial done as in as quicker time as possible with and show benefit of the drug in I guess as few patients as possible. So there is a bit of trying to stack the trial in favor of making the drug look good, and I can understand why they do that. But I can also understand why people who don't meet those criteria want to get on, and I think that's an open discussion that needs to happen. And I commend your group on being involved in that discussion. And I can assure you, the PIs and the doctors are also advocates in favor of loosening up inclusion criteria. And a lot of us spend a lot of time discussing and debating inclusion criteria with individual patients to try to get people on trials, and agree that some trials are more restrictive than they need to be. To the credit of the company, they have listened to feedback. A lot of companies, for example, and they have been brave in some other instances, leptomeningeal disease. Pretty much every other phase 1 trial I've been involved in has excluded patients with leptomeningeal disease. So, I think there are some bits that we can give them big ticks, and other areas probably need some work. I think I'm sounding like a school teacher here in terms of the report card, but it's mixed, but there are some positives there.
Well, you are a professor after all. Could you explain for us what a washout period is and why washout periods are required when engaging in clinical trials?
Yeah, so a washout period is a period where a patient who has been on another TKI or on a chemotherapy is to be off drug before they can start on trial. And the reason for that is that there is concern that if you have so you talked about your plasma levels of alectinib. If a patient has that much alectinib in their blood and then they start on a trial, there'll be potential for interaction between the TKIs. So the rationale for the washout period is to let the level of the first drug fall off, so the new drug is starting with a clean slate. But that can be tough to achieve, and in some people, when you stop the drug, the disease can take off. So a lot of investigators, myself included, try to push for a shorter washout period as possible. The original wording of this trial had a 2-week washout period. They've shortened that, but there is also an important line in the inclusion criteria that can be modified by discussion. So there is scope to even sort of reduce it further. But it also does it also does speak to I guess something that people need to be aware of. There are a whole heap of strings attached, as it were, with a trial, and in particular a phase 1. And in someone who has very rapidly progressing disease, those strings, if you like, can be pretty problematic. Those strings are there not necessarily because an investigator or even the company want there to be strings or limitations to people getting on board, but they're strings that have been put in to protect the broader group of patients, and not just cancer patients going on trials with new drugs against toxicities. And those, some of those strings are in place because of regulations by the FDA And the sort of overarching principle of that is the do no harm principle. So, so it's important to sort of be aware of that. But to your specific question about washout period, the short answer is to avoid interactions, but there is a recognition that that can be shortened by discussion.
Biomarkers in the 4 minutes that we have left. The need for biopsy or tissue or liquid biomarker testing before entering a clinical trial. Could you discuss that, please?
Yeah, so I'm a firm believer in trying to learn as much as we can from every trial and every patient who goes on a trial. And the way we do that is in part through observation of what the drug does in terms of what it does in terms of shrinking the tumor, in terms of side effects. But we can also learn from studying in plasma or tumor. And we talked about the preclinical data showing that this drug's active in, or potentially active in tumors with compound mutations. So understanding before someone gets on the drug whether their tumor has a compound mutation or a single mutation will really add to sort of database of knowledge, or sort of repertoire of knowledge of what the drug can do or what it can't do. There might be certain mutations that the drug doesn't work in. And then if we understand that, then in the future, we can spare a patient who has that particular mutation from going on a drug that will be ineffective for them. It's also possible that we can learn sort of early in terms of things like how quickly circulating tumor DNA is cleared, about how effective the drug is, and we might learn that well in advance of what a CT shows. So I think the general principle is that there's a whole heap that can be learned from biomarkers, and I'm very grateful for every patient that that consents to have additional blood draws to allow that to happen, and super grateful to those that allow biopsies to allow us to learn from that. Now, having said that, I don't think biopsies are built into this particular trial, but I know they're built into other trials that a lot of you have participated in.
Can you explain what compassionate use is and how it works?
Yeah, so look, I think it's different, different in different countries. Typically in Australia, compassionate use of a drug is not available until a drug has got through the phase 1 or even phase 2 testing program, and we know what the dose of the drug is. We know, understand a bit about its activity, but the drug is not yet available on, is not sort of approved in the country. And in that gap between when we've learned about the drug from the phase 1 or phase 2, and we know that it's safe, we know it's effective, but it's not yet available, compassionate use sort of fills that that gap. And I think that's probably the best place. So we don't use drugs that are ineffective or harmful. There are situations where potentially before a phase 1 is finished, we might sort of have enough confidence in safety or efficacy that that it can be used outside the trial. I think those are trickier, and it's sort of trickier for the companies that make the drug as well. But sometimes there are sort of possibilities of doing that, but not straightforward.
So in closing, I think I'll ask one highly subjective and non-scientific question. And it has to do with the rhetorical or the anecdotal stories that go around early on in a clinical trial, and this one in particular. Should we ignore the anecdotal stories going around, or how do you feel about how the trial is going?
So the first thing to say is that someone participating in a phase 1 trial, I think, is a supremely altruistic act. I think people do this in part for potential benefit, but I think they're super brave going on a trial. This is the first time a drug has been tested in people. We don't know what the side effects will be. We don't know how active it'll be, even if it works in tumors. In mice, I remember when I started my training, the anti-angiogenic drugs were showing great activity in tumors in mice, and someone made this fantastic line: "It's never been a better time to be a mouse with cancer." But yeah, my point is that I think participating in a trial is a big deal, and I am forever grateful for all those people that participated in the crizotinib trial, the ceritinib trial, the lorlatinib trial. A lot of them did derive benefit from the drug, but hundreds or thousands of other patients have derived benefit because they went on the trial. And I think that's an important perspective. I think if we were to judge every drug by the first dose level or the second dose level of a trial, we would throw out pretty much every drug, because we see there are we learn as we go, and different people have different toxicities, and it's by evaluating, you don't with in some ways it's like a big jigsaw puzzle, and you don't know what the whole picture is by just looking at one piece, and we need to look at the picture in its entirety to understand that. So I think the process that has been set up for trials that allows us to do that and to evaluate the results in totality is really important, and ultimately will hopefully help hundreds or thousands of people in the future.
Well, let me say thank you for all you do for all of us ALK patients. We appreciate it very much, and I enjoyed our talk today. Thanks a lot.
Thanks very much, Jeff. I enjoyed the chat too.
Want to pick it up, Mark?
Yeah, everyone can unmute and say thank you to Dr. Solomon as he spent his Monday morning with us.
Thank you.
Thank you.
Thank you.
Thanks very much.
Thank
you.
Thank you.
One more fix this quick thing when people unmuted.
Thank you.
Thank you.